As you can imagine, the intensive care setting presents a myriad of barriers in making a diagnosis due to the situation, the care situation, and the fact it's very difficult to examine children to see how they are normally. So some of the markers that would be, or features that would normally be available cannot be measured for those children. It's particularly the case with premature babies and really young children who might not have grown into a syndrome, for example. So why make a genetic diagnosis? It leads to accurate diagnosis and much better management for that child, as well as a number of additional advantages for that family in the longer term. Just to recap, the rapid R14 whole genome sequencing service is for acutely unwell neonates and children who have features highly suggestive of a monogenetic condition, where achieving a genetic diagnosis could help with an immediate change in their management. So there's been an NHS-funded service to offer this test since October 2019. And there's a couple of take-homes from this schema. The first thing is that it is a rapid test, and if you add up the days, you'll see that from thinking about a test and asking Exeter for a test, to actually delivery of results normally happens in around 3 weeks or under. The second take-home is that it's incredibly complex, and so it involves a high level of communication and coordination between different clinicians, the wards, genetics, scientists, parents, lots and lots of paperwork and blood samples, and several potential points of failure. It's an agnostic test. This means that it's a trio sequencing test comparing the DNA sequence of the baby or the child with the sequences of the two parents in a way that's not biased towards looking for particular panels of genes. What happens is the scientists look for patterns of variants that would indicate either a brand new change in the child or a recessive condition where the parents could be carriers and the child's inherited two genes, gene variants, or an X-linked condition, or perhaps uniparental disomy. And then what's really important is that the variant interpretation is done using the clinical phenotyping that has been provided by the clinicians. So this has to be of good quality. In phase 1, Nottingham proposed to use sort of 0.6 in total of a genetic counsellor, so it's a split role. And Cambridge have just been discussing that they've been using nurse specialists, again with overlap, to deliver this role, but also to support other paediatric genetic testing. In a later phase, we also have involved Leicester, who've used a third model where they have a Band 5 genomic associate, or also called genomic practitioner, to support their consultants in organising the testing. I mentioned educational sessions, and these have been really important. So we've targeted the local PICU and NICU units, as well as using the East Paediatric Genetics Forum, as well, and general paediatric talks. And those have been used to cover a large range of practical issues around the testing, things to think about, such as has somebody had a transfusion, what samples to use, and also a large number of ethical issues which are associated with whole genome sequencing. So we think that that's worked quite well. I'm just going to look at a couple of figures here, but we've had a good, looking at a snapshot from the middle of last year at Nottingham, we've had a reasonable number of referrals both from the PICU and the NICU units in the region. But we can see that from 2022, there's been a good increase in the number of referrals, and also they're on track to improve that from the first quarter data of 23. We don't have all of the data from Leicester yet, they've just got going. So here are just some examples of some of the diagnoses that have been made in Nottingham, where there are potentially the chance of changing the management there and then for that child. So, for example, that osteopetrosis means there's a possibility of bone marrow transplantation. Some conditions thought to be epilepsy have not been, etc. And also we've picked up where it was highly beneficial to screen relatives for the SN5A, for example, gene variants, which is high risk of arrhythmia. And I'm just going to focus in on FINCA Syndrome here, just to illustrate a sort of a diagnostic odyssey in which Vela Morrison really explained well when she was talking about her child, Ailey, earlier on. And this is about a 5-year-old boy who had a background of developmental delay and epilepsy, dystonia, and presented initially at 1 month of age with respiratory distress. Went on to have an absolute battery of investigations over the next 5 years. Had respiratory symptoms, hemolytic anemia, MRI anomalies found, but also some renal problems, and was referred to PICU at the age of around 5 for renal investigations. And over that time, just to point out as well, this child had had 5 lots of genetic investigations, so a huge gene panel for mitochondrial disorders in 2017, an epileptic encephalopathy panel in July 2017, a triome at Exeter, which didn't reveal anything. This was pre-R14. More genetic testing, 100,000 genomes, nothing found, 100,000 genomes, additional findings, nothing found. And then, just want to show that just after one of those sets of tests, the first publication was made of FINCA Syndrome. So it came after the Exeter trio exome had reported for the first time, so that gene was not known about. Over the next couple of years, a further few patients were actually described. We asked Exeter to re-evaluate the exome data, and they discovered that the child had 2 NHLRC2 variants, and that FINCA syndrome actually fitted very well with the way that the child had presented over those years. But what I want to say is, what we'd like in future is that the first time that child presents, they get the WGS, and so that 5-year odyssey happens in 3 weeks' time, and wouldn't that be better? That's what we want it to be like in future. That's what we're aiming for by trying to embed this service right across the region. So I just wanted to look at the national picture and compare it with what's happening more locally. This is the almost live data from Exeter. It's a couple of weeks old. So since they started doing the national testing for R14 in 2019, they've now sequenced just under 3,090 trios. And out of that, they've made around 700 diagnoses. And 400 or so likely diagnoses, and then we've got some families where we've got variants of interest as well. So if we add all of these highly significant or quite significant diagnoses together, we're coming to a diagnostic yield of 36%. So just over a third of these children have got a genetic diagnosis. So if we focus in, I've just got the Nottingham data at the moment in more detail. If we focus in on the region covered by Nottingham, so Derby, Nottingham, Lincolnshire, Mansfield, this is where we are. So we've activated 194 trios since we started in 2019, the total, and we've had 49 diagnoses. And that actually equates to 26% pickup rate. We've got a number of variants of unknown significance for some of our families, and we've got a few that are still in the pipeline. Where are we going in future? I've got 35 seconds. We're in the evaluation phase. We're doing cost-benefit analysis. We're doing a vertical audit. We're looking at how to embed services. We're looking at efficacy, equity of access, how many tests we're doing across. We're about to start a virtual board round to say, can we get those children on NICU, PICU, and to have genetic test earlier? Has it been thought about for those children? I've got 10 seconds. I'm going to thank everybody. Okay.